Hi, everyone. Welcome to week 3 of Scientist Chat. Thank you for joining us today. Just a few guidelines again. I please ask that attendees under the age of 13 that they be supervised by a parent or guardian, as well as just to let everyone know that this session is being recorded. So once again, the reasoning behind this is due to the Let's Stroke Science Challenge. It's a weekly quiz. This week, which is the third week of the quiz, the quiz will be taking place on April 30th, which is Thursday, and we will have an English quiz at 10 to 11 a.m. as well as Eastern time, as well as 1 to 2 p.m. Pacific time. There's also a French quiz which is offered as well, beginning at 2 to 3 p.m. Eastern time. So this week, our topic is physics. So the topic that'll be covered in the quiz this week centered around light and its properties, reflection and refraction, as well as how we see. So as usual, you can log on to the Let's Talk Science website and follow the link to the Let's Talk Science Challenge, which will lead you to the handbook that pretty much covers the topic that would be tested on the quiz. Well, that'll be quizzed on Thursday. Okay. So just a few things I'll be looking at is things like light and its properties. So we're looking at the crest and the waves of light. So we're looking at things like the crest, the trough, and the wavelength. We'll be looking at the various parts of the electromagnetic spectrum and the primary colors. In terms of reflection and refraction, we'll be going over the principles of reflection and refraction. So we're looking at the incident light and the reflective light, as well as the normal, and the impact that the smooth versus diffused versus rough surfaces have on the reflection that is produced. Additionally, we'll be looking into how we as human beings see, which is a very interesting topic as well. So it'll all be covered under the quiz on Thursday. So without further ado, please join me in welcoming Dr. Michael Kolios. He's a professor in the Department of Physics at Ryerson University. He's the Associate Dean of Science for the Research and Graduate Studies. And his research interests include the ultrasonic imaging, acoustic microscopy, the use of microbubbles for imaging and therapy by heat transfer, and optical coherence tomography applications. So without further ado, please join me in welcoming Dr. Kolios.
Okay, thank you. So I'm just gonna try to share my screen right now. Thank you very much, everybody, for participating. And I do apologize if my eye gaze is in different areas in space, as I have multiple screens and an actual camera. So what I'm going to be talking a bit today is about light and sound in medicine, since the module that you're covering in Let's Talk Science has a light theme. I will be talking about a little bit about the research that I do in this area. And even though there's a lot of different aspects of my research program, I'm going to be only focusing on a couple of examples, because otherwise it would be kind of unwieldy. So as I say with my dad jokes here, a great STEM ecosystem, right? Because math and science are used very much in the applications of light and sound in medicine. So just a second. A little bit about myself before I start, just to let you know how I got interested in science and why I chose the topic I did. While I was born in Toronto, I went to Greece with my parents when they moved back. So I finished my middle and high school in Greece. And that little icon you see here right beside is actually the goddess Athena, which is the goddess of wisdom and mathematics. So I guess that was kind of an omen that I'd be ending up in a STEM-like subject. I was interested in math, I was interested in physics, but I really didn't know what I wanted to do when I was in high school. I entered the Bachelor of Science in Physics program at the University of Waterloo, since I had a predisposition towards physics and computer science. But even then, I really didn't know what I wanted to end up doing during my bachelor's. But it was really in the third and fourth year of my degree where I really got the research itch, so to speak. It was an experience I had in Atomic Energy of Canada as a summer student once, where I saw I could actually contribute to research in a fourth year thesis project that really made me think, okay, maybe I could hack trying to do a PhD, which was my next step. So then I went to the University of Toronto. I completed my PhD in something called medical biophysics, which really talks about the applications of biophysics in medicine. I'll talk a lot more about that in the next few slides, so I'm not going to belabor it too much. And then I joined Ryerson. I joke about it, one PG, one pre-Google, since Google was formed in 1997. So yes, there was life pre-Google, and that's when I joined Ryerson University as a professor. So that was kind of my path. Now, what is medical physics? So actually, maybe you do know much about it, maybe not. But basically, it's all applications of physics in medicine. In fact, there are many organizations, and the International Atomic Energy Agency had as the International Day of Medical Physics, the 7th of November, 2019. That's a special date. That's Marie Curie's birthday. Right? She's a scientist that did a lot of work on radioactivity and is one of the few individuals that got 2 Nobel Prizes, Marie Curie. So now let's think about medical physics again. What is it? How really are physical concepts applied to medicine and biology? First is therapy. And I highly doubt most of you know who this is. This is Sylvia Fedoruk. She was a scientist, I think back then at the University of Saskatchewan, and she played a seminal role in the development of the first radiation machines. So these are the kind of instruments that are used to treat cancer, because radiation can treat cancer if it's applied very locally at the appropriate location where the tumor cells are. So medical physics really had its big bang, so to speak, in the applications of physics in medicine related to cancer, because they saw not only can you make images with X-rays or with radiation, but you could also, if the dose is high enough, actually treat cells. The other big application in medical physics is imaging, of course. Here you might recognize what these are. These are images of the brain. And these are done with something called magnetic resonance imaging. And to me, magnetic resonance imaging is kind of one of the amazing examples of the integration that you need in the STEM sciences to be able to do this kind of high-tech technology. You have to know a lot about the chemistry. You have to know a lot about the physics. You have to know a lot of computer science and math to be able to reconstruct images. So these are kind of the 2 main applications of medical physics, therapy and imaging. In fact, the contributions of Canada to the field are very significant. What is called the cobalt bomb, which was one of the first radiation machines that were produced, was developed by Sylvia Fedoruk and Harold Johns in the 50s. And you could see here Princess Margaret visiting the Princess Margaret Hospital in Toronto many years ago when they were kind of demonstrating the first such device. And you've made it big when you get a stamp, right? And here is the stamp that commemorates the development of this radiation technology. This is also known as the cobalt bomb, still used in many parts of the world, but we have more advanced technologies now in Canada, so you won't see it really in Canada. It's a very strong program. In the Department of Physics here at Ryerson, we have something called the Biomedical Physics Masters and PhD, which is accredited by something called the Commission on Accreditation of Medical Physics Education Programs. So medical physics as a discipline is a discipline on its own, and you could get a graduate degree whose courses have been part of the approved by the accredited process. And there's an actual job description at most cancer hospitals, that being called a medical physicist, and those are the individuals that are responsible for trying to figure out how to deliver the radiation only at the location of where the tumors are. But you can see here light, right? These are kind of just light. This is actually from my lab, I think. These are kind of laser labs that we have. So I'll talk a little bit about light now. You probably don't know this, but in 2015, it was the International Year of the Light. Now, you would say, well, Prof. Kolios, that's like 5 years ago. Why are you showing this? Well, because the website that they have here, light2015.org, is still live, and you could go and visit it. And I highly recommend you do if you're interested to find out more material compared to what's already been shared with you through Let's Talk Science. And you can see here the kind of tabs, and each tab has actually detail. So for example, you could learn about lasers, you can learn about light and life, light and nature, art and culture, kids and optics, etc. All of these are little like tiles that you could click on, and it gets you to that material. You can learn about the history. So a lot of people, don't know who what is sometimes called the father of modern optics is, which makes 1,000 years ago, he developed a 7-volume treatise on optics. So he was kind of one of the first people to understand really the nature of light. So again, I would highly recommend it. Excellent website to go visit and to get information should you be interested in learning more about laser light. I focus much more on something some people would call biophotonic. So that's kind of the applications of light in biology and medicine. And there's just many of them, way too numerous to list. But I guess one of the big things is in imaging, right? So, using light to be able to infer things about the physiology and the human structure of tissue. A big boost was when the laser was first produced. I mean, you can see, I hope, the red dot that I'm sharing here. It's not a real laser pointer. It's, I guess, a fake laser pointer, but still it shows you kind of the impact that lasers have had here in many different fields: dermatology, ophthalmology, dentistry, etc. In fact, even if you like tattoos, right? Tattoo removal is done by lasers many times, etc. Many, many different applications. And if you really want to push the applications and how recent the applications are, due to the COVID crisis now, what has been a very, very hot seller is something called an oximeter, right? The oximeter basically measures the oxygenation status of the hemoglobin in your red blood cells. This little device here, if you actually open it and look inside, they have what is probably an LED laser inside. So basically they use 2 different wavelengths of light to look at how much oxygen is bound to the hemoglobin in your red blood cells. So again, as you can see, light permeates everything, even during this COVID crisis, because one of the things that a lot of people have noticed is before somebody has problems with the lung, they see a big drop in the oxygen content of red blood cells. So now I'm going to go a little bit more to my research, and hopefully not more than 5 minutes. So I do a lot of work on something called photoacoustic imaging. So what you see here is you see a picture of lightning, right, hitting a structure that most of you should recognize here, which is the CN Tower. So you can see here that lightning is striking the CN Tower. Actually, it hits it at least 60 to 100 times a year. There's a lot of people that actually have their research labs up here because they study lightning science. But that's not what I want to talk about. I want to talk about something called photoacoustic imaging. So you know that when you see this lightning bolt, because you studied your physics and you know that the speed of sound in air is approximately 300 meters per second, for every 3 seconds you wait after you've seen the light, if you hear the thunder, that means it's about 1 kilometer away, right? 3 times 350 approximately. So you see the lightning. If you hear the thunder after 3 seconds, it's approximately 1 kilometer away. If you hear it after 6 seconds, 2 kilometers away. If you hear it immediately, time equals 0, you're toast. That means the lightning hit you. Anyway, so why does the lightning actually produce sound? Well, it's fairly simple to conceptualize. The column of air right beside the lightning gets heated up, it expands. That expansion creates the pressure waves that we hear with our ears. So that's really the basis of photoacoustic imaging, right? It uses light. That light gets absorbed. The absorption of light creates a pressure wave, and then we detect that pressure wave. So you do that every time, in a sense, you hear thunder after the lightning. So how do we do this in real life? Well, you have something called an ultron transducer. Some people might have seen these devices at hospitals. It's something you can hold in your hand. And in a way, you could think of that only as a speaker-receiver kind of thing, right? So, it's something that, a transducer, something that can emit sound if you want it to, right? Because that's how ultron imaging works. You send sound and then you receive it. But in this application, in photoacoustic imaging, it's the light that creates the sound. So that's just sitting there listening. So if you think about it, it's kind of like an ear, right? Listening to any sound that's produced. So you have a pulsed laser source typically. Here's the light. The light gets turned on. It heats up whatever absorbs the light ever so slightly. So these temperatures are like 0.01 degree Kelvin, very, very small temperature increases. And then the pressure wave just travels all through space, right? And here is you have your transducer, the thing that actually listens to the sound. So basically, in a sense, what we're doing is the same thing as thunder and lightning, right? We're heating up something, that thing that heats up expands ever so slightly, and it produces pressure waves. And we just detect those pressure waves with the ultrasound transducer technology that we've developed in the past. And then the cool thing is that all these algorithms and all this instrumentation that's been developed for ultrasound, we could use it to make images on an ultrasound machine. So as long as you could synchronize the laser with the ultrasound, then you can make images of whatever absorbs the light. Now, you know, Adisha talked about the electromagnetic spectrum. You could use any type of light to produce these kind of photoacoustic waves. We typically use something called the near infrared spectrum, this area here, and that's because light can penetrate a lot more through tissue in these wavelengths. You don't believe me? Well, if you have a green laser pointer and a red laser pointer, you could put it against your finger, and you will see when you use a red laser pointer, your whole finger will light up red. Do it in a dark room or darker room and you'll see it. You put a green laser pointer and you tell me how much you see from that. Okay? So anyway, you could use any part of the wavelength. If you use radio waves, so you know these longer wavelengths, you could get what is called thermoacoustic imaging. If you use this part of the spectrum, X-rays, you could do something called X-ray acoustic tomography. Okay? So again, you could use many different types of part of the electromagnetic spectrum. And interestingly, the sounds they make, so you know, you've heard thunder, so you know what thunder sounds like. When you do photoacoustic imaging, the type of sound you hear is the same as the type of sound you get from bursting balloons. Now, I won't go into the physics of that, right? But it's very interesting that the type of sound you get is the same sound you get as bursting balloons. And we have these very complex devices, right, that would listen to the sound. So this is one of the instruments we have in our lab. This looks like a microscope, a modern microscope, right? And it really actually is. And all we've done is we've changed the top part and put a transducer here to listen to the sound. So we use the same part of the optical microscope as you would normally to shine the light on whatever it is here that we want to listen to the acoustic waves from. And then we use this transducer just to listen, right? So the transducer is just sitting there, and every time we pulse the light, we hear a sound. And this snap I'm doing is kind of like the sound we hear. Well, we don't hear it because it's ultrasound, but the types of sounds that are produced when we do photoacoustic imaging. And here in our device, we have many different optical wavelengths. And we can listen to many different types of sound, right? Different frequencies. All of these, of course, very high frequency. No way on earth you could hear it. That's why you need a specialized instrumentation to detect it. In fact, this is the red blood cell right here. You could probably see it in the image. And I'm going to show you, hopefully this video will work. And what's going to happen in this video is this red blood cell is going to rotate. And as the red blood cell rotate, you're going to see how the sound changes. This is the sound, right? You can't hear it. This is a visual representation of the pressure wave. So what's going to hopefully happen here, oops, let's see. I guess the laser pointer inactivates a bit the video. So let's just play the video. You can see the red blood cell rotating and the sound totally changes. So it's interesting. This red blood cell produces those photoacoustic signals, and we're listening now to the sounds made by this red blood cell. And you could see here in the frequency domain, so these are the different frequencies of the sound that are produced, very, very different frequencies depending on how it's oriented compared to our device that listens to that sound. Now, this is actually the blood of one of my graduate students, right? But you could, seriously, when you're in grad school, I really rely on the blood and sweat of my graduate students, kind of literally in this case. Anyway, so this is the red blood cell. And now if we go to the next slide, I just want to show you an application and just a couple of more slides. So, and I'm mentioning this because it's very recent. So the publication is going to come out in a few days, and there's going to be probably a press release on this. But one of the problems people have, and you're going to see another application here, is by looking at kidneys to determine whether they're scarred or not, right? Whether they have a lot of fibrosis. So if you look at the ultrasound images here, and you say, can you see the difference? Maybe not, right? It's not that obvious, and certainly not very quantitative. This is a healthy kidney that has no fibrosis. This is a kidney that's scarred and has significant fibrosis. So if you just use ultrasound to look at this, you can't really tell. But if you now use photoacoustic imaging and you use 2 different, or 3 different in this case, wavelengths, so 3 colors of light, because the collagen that is produced during the fibrosis absorbs light differently, you can estimate how much collagen there is in the kidney. And then you could produce images that now look like this. So you could see now in this photoacoustic technology, this is the healthy kidney that doesn't have that much collagen or fibrosis. This is the kidney that has a lot of fibrosis. So this is the first time you can really get these kind of detailed maps of fibrosis. Again, the application is light. You send the light and you listen to sound that comes back. And this is the final image. This is from a group at Caltech, which shows now in a mouse, you can see this is what is called the imaging plane. So they're making now photoacoustic images as they scan down the plane. And you could start recognizing now many different parts of the body that are highlighted here, right? Here's a zoom of whatever's in this region of interest. You could see the kidney. You could see, it's real time, right? You could see the intestine, the spleen, the muscle. Now, because what absorbs light mostly in your body is red blood cells, this is a map pretty much of the human vasculature, right? So you can get these vascular images with great detail. And again, this is just by listening to the sound that is produced by the light. So I would like to thank you for your attention. This looks like a sunset and light, but it has nothing to do with that. It's just ultrasound scattering from a bead. I hope you enjoyed my little presentation, and I'd be more than happy to answer any questions.
Wow. Oh, this
I'm going to try to stop sharing for now. And,
yeah.
Okay, there we go.
Oh, very, very, very interesting presentation. I definitely learned a lot. I hope everyone in the audience learned a lot as well. So I'll invite the audience members to please insert your questions in the chat. In the meantime, I will be asking Dr. Kolios some of the questions that came in ahead of time. Is that okay, Dr. Kolios? Okay. So the first question that we got is how do bubbles form?
Good question. So one thing I did not talk about here, right, is I do a lot of research also on making bubbles, because bubbles tend to be very good what is called ultrasound contrast agents. So in other words, again, a little bit technical, but I'll try to explain. If you look at a kidney, let's say, if you look at a liver with ultrasound, you really don't see that much detail. You just see kind of like a little speckly pattern or something like that. So you have to put something in there to start seeing things like the blood vessels. And one of the things people put in is microbubbles. Now, these microbubbles have to be very, very small, right? Because you don't want the bubbles to be blocking an artery or some vessel, right? Because that is basically a stroke. That's bad. But if you make the microbubbles about, smaller than a red blood cell, 2 to 3 micron in diameter, they could just go around in the body, right, for as long as they last. So typically to make these bubbles, there are 2 approaches that we use in my laboratory. One of these approaches, and that's based on a collaboration we have with Case Western Reserve University in Ohio, Cleveland, is basically we use lipids, a solution of lipids, and we mix it with gas and then shake, right? Like shake and bake, so to speak. A little bit, again, I'm greatly oversimplifying the process, right? But with a process called mechanical agitation, you could create these bubbles. The key here is trying to make them stable, because as you've seen, with 7-Up or things that just fizzle away, right? Those bubbles, if you don't make them carefully enough, don't last very long. So, and the other way we do it is something called microfluidics, but I won't go into too much detail with that because it is a bit technical, but ultimately we just kind of try to think of, blowing bubbles with a straw, scaling it down and doing it with specialized devices.
So how long an average would one of these bubbles last
Oh, that's a
within the
very,
body?
yeah, that's a very difficult question. I mean, that's
Okay.
why I have my collaboration, right, with the person at Case Western. I'm not a materials science expert, but she has made bubbles that last much longer. Because typically, when you inject a commercial product called Definity, you'd be lucky to get good echogenicity or good signal for a couple of minutes, right?
Okay.
And that maybe is a little bit too short for a lot of the applications that we want. So the bubbles that I'm talking about could last about 10, 15 to 20 minutes. But again, there's just so many little details, right, that it makes the answer it makes the question very difficult to answer with great rigor or great detail. Yeah.
Okay. Okay. So the next question that we had ahead of time before I go into the chat was a question about photons reflecting on a mirror. Let me see if I can find it properly.
Okay.
The question is, if there was a photon bouncing between two perfectly parallel mirrors in a vacuum, how hot would the mirrors be?
Oh boy. Okay. So I guess that's a hot question. Let me think about that a bit. So what you're kind of describing is an optical cavity or resonator, right? So I will qualify by saying the answer I give is hypothetical, right? It's a very theoretical, contrived system we're describing, but I'll just give you a estimate, right? I would say very hot, right? Because if it's a real vacuum, the loss of energy of the photons as they bounce back and forth between the mirrors, right? I would assume would only be where the mirrors are, right? That's where you'd have the exchange of energy as the photon interacts with the mirror. If it's highly idealized, then there's no way for the heat to dissipate, right? So, I don't know what kind of real life scenario that would be, but if there was no heat dissipation, then all that energy would be confined into that space and presumably would get very, very hot. But I think in real life, what happens is as the mirror heats up because of the multiple reflections, heat conduction, right, provides an upper bound, right, for the temperature that would reach. Again, I'd have to think about that much more carefully, but that would be my first guess.
Yeah. Okay, perfect. Thank you. Okay, so the next question coming from the audience here is, do higher energy photons give off more entropy than lower energy photons?
Give off higher entropy. So I'm just going to answer this maybe in the context of photoacoustics, the work I do, right? So you have photons of different energy, and that different energy corresponds to a different color. And that different color interacts differently with the different biomolecules in your body. So in other words, remember that example I gave you of the pulse oximeter, right? They had something that would go onto somebody's finger, right? How does that work? That works because you could use two different wavelengths of light, okay? One higher energy photon, let's say one lower energy, right? Two different wavelengths. Mm-hmm. And the hemoglobin, so each red blood cell has about, 10, 15 million hemoglobin proteins in it. So it's like stacked with hemoglobin, right? That's what the oxygen binds to, right? And that's why so much oxygen could be in the blood. So anyway, when the oxygen binds to the hemoglobin, right, it changes how much light gets absorbed at particular wavelengths. So in other words, when you use high or low energy photons, right, the absorption you get from the red blood cell changes. And by measuring that, you could estimate, right, the oxygenation status of the hemoglobin in your red blood cells. So that's how the pulse oximeter works. And in fact, in photoacoustics, we could do that in depth and we can make images of that. So in the pulse oximeter, you're really just getting an average value for your whole finger, right? You're not looking at a single vessel and saying, how well oxygenated is the blood. So with photoacoustic imaging, though, because you can make images of that, you could even pick blood vessels and say, okay, this one has blood that's well oxygenated and this one doesn't. And one last thing I'm going to say about that, it's amazing work, really. I mean, you could do that in the rat brain, and you could do something called the whisker test, where they kind of tickle the whisker of a rat, and you could see which parts of the brain get activated, and you could see the change in oxygenation status of the vessels that is created by the change, the physiological change related to the excitation.
Oh, wow. That's pretty cool. So the next question here is, so a few of this audience members want to know a bit more about the eye. So one actually is asking, how does the eye work? And there's a second question, it's a bit more technical, that's asking about, is it possible to transplant genetically modified photoreceptor cells in a human eye so they can see other types of light that cannot normally be seen?
Wow. Okay. So again, not an eye expert, but I'll tell you the basics, right? You know how I understand it. I mean, basically, what your eye is like a little bit of a camera, right? So you have your eyeball, right, where the light comes through. You have a transparent kind of membrane, and the cornea that you have in the front, and all the action really happens at the back of your eye, the retina, where the photons interact with cells that have pigments that absorb the light and create electrical pulses, right? So again, I'm oversimplifying a bit. Those electrical pulses basically, the way it works is you have light. That light is refracted through the lens of your eye in the front. It's focused in a complicated way in the back of your retina, right? And then it produces electrical pulses that you then interpret as an image. So, for example, I'm older now. I need these things, glasses, right? If I don't do this, now you're all fuzzy. That's because now the photons that go through don't focus on the retina, and therefore it's hazy. I need extra glass to focus things on the retina. So really, when we talk about vision, it's a combination of physics, but also how the brain works and how it interprets the images. Because if you look at the images of the neuron, theoretically, those images are inverted on our retina, right? Because of the ray optics and how it works, but we interpret it upwards. So the second question, I think, was about somehow doing genetically. So can you repeat that about genetically modifying something?
Definitely. So the question says, is it possible to transplant genetically modified photoreceptor cells in a human eye so that they can see other types of light that you cannot normally see?
Okay, so in principle, I can't see why not. In principle, I've never heard of this, first of all, right, for humans. And I would say good luck trying to get a REB or Research Ethics Board to approve such an experiment, right? I mean, that would be very, very difficult. But ultimately, it's about, as you identified, cells producing pigments that absorb light that produce electrical signals, right? So, in just a very kinds of principle approach, I cannot see why that cannot be done. Practically, though, I can imagine that it's excruciatingly difficult, I would think.
Okay.
Again, I'm not a molecular biologist, so I don't know how difficult it would be to do this transfection. I don't know what kind of cell, I don't know what the approach would be, basically.
Yeah, that's a good answer. Okay, so following question is, can myopia be healed?
Can what?
Myopia?
Yeah.
Can it be healed?
Healed. Well, again, I mean, the way you accommodate for the myopia is, glasses, right? Things that focus the light in a different way. Another way of changing kind of the refraction of light is by doing surgery, right, on the eye itself, right? So there are that's how lasers are used now many times too, right? Lasers are used, to change, in a sense, the shape, right, of the surface of the eye so that the light gets focused where it should be on the retina. Now, about myopia, though, I'm not really sure, right? If that is one of the indications that can be treated with LASIK or those kind of laser eye surgeries, right? But certainly, certain types of optical pathologies or not being able to see well can be healed or corrected is a better word, I think, right? With those kind of procedures. It's very interesting. It's a relatively new technology, right? And I really would have loved to have seen the first people that thought about that idea of, shooting lasers to the eye to change the surface of the eye in a way that corrects for the focusing of light. Very crazy.
Yeah, that's yeah,
but it works.
Yeah, very, very interesting piece of science there. It's always interesting to see like people who do these groundbreaking technologies. What were they thinking?
Exactly. What were they thinking?
Yeah. Just something in a dream or something. I don't know.
Yeah.
So I have a question here. So you mentioned photoacoustics, correct?
Yes.
So I have a question here about something called sonoluminescence. Is it a similar thing, different word name for the same?
Very different, but let me explain it just very briefly because it is also related to bubbles. Okay. So what is sonoluminescence? Well, now imagine you have a bubble, okay, and the bubble has a gas in it, right? And the bubble has a certain size, okay? What do you think would happen, right, as you decrease the volume of the bubble? Make it smaller, right? If you think about, and again, you probably haven't covered it, I'm not sure, the ideal gas law, like PV equals nRT, okay?
That's probably the chemistry sections.
Chemistry section, yeah. So what you'd expect eventually is an increase in temperature, right? So as you increase the pressure, right, you're going to be increasing, the temperature. So when you're now using ultrasound to make microbubbles oscillate, and you use a strong ultrasound field, right, you're compressing gases very fast many times. So what happens is you get the formation of high temperatures, especially when the bubble implodes. So in some cases, you basically break the bubble, it implodes, right? That inertia was called inertial cavitation. So very theoretically, some of the temperatures reached there during this process could reach, 5 to 10,000 degrees Kelvin. These are extremely high temperatures. So it ionizes the gases, and that ionization produces light. Okay, so sonoluminescence is the light that is produced typically when you have, ultrasound incident on bubbles with the right frequency and the right pressure to induce very high temperatures to make the bubbles produce light. It's actually fascinating to see because if you do everything right, and it's a difficult experiment, you could see pulses of light that correspond to the bubble oscillation, right? So light. So that's sonoluminescence. And, one last thing I'm going to say, which is very interesting, there was a paper in Nature, which is kind of, I think it was Nature Science, I can't remember now, which had shrimp sonoluminescence. Shrimp? What is that? I mean, there are certain types of shrimp, I think they call pistol shrimp, that have a claw that goes, right? And that claw makes a noise. And initially they thought that noise was the claw touching the two edges, but it's not. The claw, when it closes like that, makes a bubble, and then the bubble explodes, and the bubble exploding is one making the noise. But what they noticed is when the bubble exploded, it was sonoluminescent, so it produced light also. Yeah. So shrimp could do it also.
Okay. Wow. Wow. That's so is it because of the speed at which they do the?
Yes.
Oh, okay.
Yes. Very, very rapid, right? Very rapid closure, creation of the bubble implodes, very high temperatures and pressures. You get the ionization of the gas and light. Yeah.
Okay. Wow. Very, very cool. So I have take around 2 or 3 more questions here. So this one is just going back to the electromagnetic spectrum. The question is, why is the electromagnetic spectrum called the electromagnetic spectrum?
Right, because again, I will give you my guess, right? But if you think of the nature of light also, right, in itself, what is it? After many experiments in the 18th and 19th century, what they found is that basically it's sinusoidal oscillations in the electric and magnetic field in space, right? So in a sense, when we talk about the wavelength of light, right, we're talking about the distance between two peaks of that oscillation. And it's what is it the oscillation of? It's the oscillation of an electric field and a magnetic field, right? So you probably saw kind of like traveling waves or waves, right? And you have the electric field on one axis and the magnetic field on the other, right? So by nature, a lot of this, energy is transmitted through these electromagnetic fields, and thus it's called the electromagnetic spectrum.
Okay. Right. So I have two more questions here. They're about the work that you did with the blood cell.
Mm-hmm.
So the student wants to know first of all, how did you isolate that blood cell, and also why does the blood cell produce a lot of sound?
Correct. So I'll tell you a funny story too about how science works about this also. Sorry, the first part of the question was?
How would you isolate the blood cell?
Right. So that's a normal procedure, right? So I mean, you could take blood and you centrifuge it, right? So you put it in something that spins a lot, right? And as it spins, the liquid part of the blood separates from the non-liquid part of the blood. In the non-liquid part of the blood, there are the red blood cells, and then there's something called a puffy coat. There are white blood cells, right? So, you could go to where there's only red blood cells, right, and isolate them. It's not a difficult procedure in general. In this case, however, I'd have to so in the data that I showed, I'd have to double check how that experiment was done, because I don't think we did anything very complicated there. That was just a finger blood prick, right? And then basically we diluted it enough so that in the field of view of the microscope, right, the only thing that we saw was a red blood cell. And then we put it actually in a gel. And it's interesting. You saw it rotate. The rotation was because the cell was falling in the gel. It hit the glass slide, and then it pivoted, and it fell down. And we were able to capture all that using the photoacoustics, right? And the physics worked beautifully. Just shows you how mathematics and physics, for whatever reason, right, could be used to describe nature like that. So again, sorry, the second part of the question was, Adisa?
So the second part is, why does the blood cell produce a lot of sound?
Good question. And that's the funny story, right? So it'll get a little bit technical, but when the sound has such a high frequency, 1 gigahertz, let's say, the attenuation of the sound, so in other words, how much the sound gets reduced, right, is huge, right? It's many hundreds of dBs per centimeter or millimeter. So the people that made the device, when I told them I wanted to do this experiment, they said, nah, it's not going to work. They had a German accent because they were Germans, and, it kind of made me very sad because I thought, okay, should I put all this effort to do something where the makers of the instrument tell me it's not going to work? But I did some other calculations, and what I found is that there's going to be a very strong photoacoustic signal produced, and therefore that attenuation, even though it's huge, the signal produced was huge to begin with. Now, it goes back to the question that was asked. Why was this signal huge to begin with? And that's because the light is absorbed by the hemoglobin. So the question is, in a red blood cell, are there many hemoglobin molecules? And the answer is yes. As I said, there are tens of millions of hemoglobin molecules inside a red blood cell. Each and every one of them, in a sense, absorbs the light, and therefore you have the production of a instantaneously, temperature expansion, and then the red blood cell, even though it's so small, right? Because it has so many hemoglobin molecules, it produces a sufficient loud enough signal, so to speak, for our devices to hear it.
And I have one more question for you. I think we'll wrap it up with the question that we asked all our guests. So the question is, because of this time of the coronavirus, how have you, as a scientist, and some of your colleagues adapted to deal with these new situations?
Yeah, so obviously it's very difficult, right? And scientists, well, I could tell you because I'm associate dean too, it impacts scientists very disproportionately. So imagine you're a mathematician, right? Or you're a computer scientist that just works on algorithms. The impact to your research program something like this would have is not as great as if you were a laboratory scientist. So a lot of my work is experimental, like that microscope I showed you in the blood, etc. Right now, the facilities in my research lab, I cannot access because there are restrictions. So anybody who does research that is very dependent on experimental apparatus, right, is very highly affected. So let me focus now on that subgroup, the people who are in labs and therefore are not able to do the research that they can do. Well, I've asked them to do several things. And one of the things I'm asked them to do is, first of all, go read the classic papers, again, more carefully. So in science, there are some papers that are cited a lot. So you could consider those as the classic papers. And even though I tell my graduate students and members of my lab to read them, I know that human nature is such that when you read them the first time, you get some of the concepts, but you don't get all of them. But if you've worked on the problem for a year and then you reread the classic, you will grab so much more material. You'll understand it, right? So that's one thing. The other thing I've asked them to do is to do simulations, right? To do what the mathematicians, let's say, would do, or the computer scientists. So in other words, okay, you don't have access to the instrumentation, right? But are there ways that you could simulate that so that you could sit in front of your computer, right, and do some of that work? Of course, that's not easy, right? Because, you have to learn how to program or whatever the approach is. But the fortunate thing is that many vendors and many companies now, if you look online, are offering webinars, right? So that people get familiarized with how to use these technologies. So we're very fortunate that many industry leaders, but also societies, right? Like scientific societies, are making a lot of webinars available to the research trainees that I have. And I've definitely asked them to do a lot of that. And the final one is writing papers, like putting something on paper. I'm a very, very, very strong advocate of. You really, if you don't put it on paper, and it doesn't have to be literally paper, right? It could be a Word document, right? But if you don't try to articulate those ideas in a rigorous way, in an organized way, you'll never be able to explain what you want to explain or understand what you want to understand as well.
Okay. Wow. That was pretty good advice, even for myself. I think I'll take away some of your advice there as well.
I'll send you the email I send to my group, which has about 10 suggestions.
Perfect. Please. That'll be much appreciated. Yeah. Okay. So I just want to take this time to thank you very much for joining us today, Dr. Kolios. I want to thank the audience for being here. Just a little, for those whose questions were not answered in this live chat, we will be collecting those questions and sending out the answers to you in a follow-up newsletter. Okay. So once again, thank you, Dr. Kolios.
Okay. Thank you very much. Okay. Yep. Bye now. Bye, everyone. And thank you for participating. And stay interested and curious in STEM. Bye now.